I'm just going to talk about T cell dysfunction because Adrian actually gave a great review on B cell dysfunction. And so as you know, HIV and HCV infection are examples of chronic virus infections where during the acute phase of the infection, you can see quite a large amount of virus that then decreases by many logs of magnitude. So you have an incredible actual control of the infection during the acute phase by the immune system. But the infection is not completely controlled, so something's happening. And we think that the major effector cell that's controlling virus throughout the infection is the cytotoxic CD8 T cell. But so there's a battle going on between the virus and the CD8 T cells, where the virus eventually seems to subvert that immune response. And the way the virus subverts the immune response is most likely through, it's just escape from the epitopes. So if you look at every patient during the early stage of HIV infection, you will probably find about 3 to 5 T cell CD8 epitopes in which the virus has escaped by mutating from that epitope. But the curious thing is that if you look at every patient with HIV infection, you will find CD8 T cells that respond to HIV epitopes that haven't mutated, that are actually invariant. And if that's the case, why is the virus not controlled? So there's something happening with these CD8 T cells that I'm interested in discussing, because these cells should be controlling the virus. So when you measure function of CD8 T cells, the way that immunologists have been measuring them is sort of a archaic way, because in human immunology, you have to take the CD8 T cells from the body and study them in vitro in a test tube. And so the way that they're studied, their function is studied, is you take the CD8 T cells and co-culture them with an antigen-presenting cell that's pulsed with peptide or transfected with a gene, and you measure proliferation, survival, cytokine production, chemokine production, effector enzymes such as granzyme and perforin, degranulation capacity is measured by CD107A expression, and killing ability of the target cell. And traditionally, immunologists have said that if the CD8 T cell has all of those functions, it's functioning properly. But I think that may not necessarily be the case, in that maybe depending on the state of maturation of that CD8 T cell, only a subset of those functions will be present. But then that confuses our interpretation of what is a dysfunctional CD8 T cell, and you have to keep that in mind when I'm giving the talk. So classically, in the mouse model, the LCMV mouse model was the first model to be shown in 1993 by Zinkernagel's group that T cell dysfunction occurs. And what's happened is that if you give a high amount of a certain strain of LCMV, if you give a low dose of a certain strain of LCMV, the virus is actually cleared, and it's cleared because you can see a good CTL response. But if you use the same strain and use higher amounts of virus, then all of a sudden the virus isn't cleared and the animal dies of immunopathologic disease. And when you look at that animal, you actually do see an initial CTL response, but then the CTL response disappears. In addition, what you see in this situation is that you may see CTL responses to different epitopes compared to the epitopes that were controlling the virus with the lower dose of virus. And you see two types of dysfunction, at least, in these CD8 T cells. So this is a tetramer stain looking at some LCMV-specific cells, and you can see that in some, you actually can detect tetramer, and yet when you measure cytokine production, you can't see cytokine production in those tetramer-positive cells. In other epitopes, you actually can't even find the tetramer, so the CD8s have been completely deleted. So you see two things. You see just dysfunction of cytokine production, or you actually see loss of those CD8s to another epitope. The other interesting, curious thing about this model is that if you deplete the CD4 T cells in these mice during the infection, you can exacerbate this dysfunctional T cell response. And this is shown here, that if you give LCMV by depleting the CD4 T cells, you can actually get a more progressive state T cell dysfunction. So this state of T cell dysfunction has now been coined T cell exhaustion, which was probably coined by John Wary. And essentially what happens is that when a CD8 T cell sees the virus infection, it becomes an effector T cell. And if the effector T cells actually are successful at clearing the virus, the effector cells become quiescent memory cells. And when you take these quiescent memory cells from the mice and look at them in vitro, you'll see that they do all the functions that I listed before. However, if you have a chronic persistent virus infection and you take the CD8 T cells and look at them in vitro, you'll see varying degrees of, quotes, dysfunction in these CD8 T cells, from increased apoptosis, and you start seeing decreased ability to proliferate and produce IL-2, followed by TNF-alpha and interferon gamma. And it seems like the more virus you have in these animals, the more dysfunction you see. Now, in HIV infection, well, first of all, we know that CD8 T cells, in order to really function well, they need the help of CD4 T cells. And the way that happens is that CD4 T cells upregulate CD40 ligand when they see antigen presented by the dendritic cell. And then that dendritic cell, after being signaled through CD40 ligand to the CD40 receptor, becomes hyperactivated by increasing MHC class II expression, producing cytokines like IL-15, and this allows the CD8 T cell to be primed maximally and probably to have a better ability to function. So in HIV infection, it's sort of a scenario for T cell exhaustion because during CD8 priming of the virus, the CD4s are probably already being deleted that are specific for HIV. So we see loss of viral specific help early on. We see high antigen loads. And when you take CD4s and CD8s from HIV-infected individuals, you see deficiencies in proliferation and cytokine production. In addition, you might also see altered immunodominance, where some epitopes get deleted during the course of the infection. So there's been a lot of work looking to see what molecules are upregulated that are associated with this T cell exhaustion phenotype. And there's a whole collection of signaling molecules that are expressed on T cells that are important in signaling up both positive and negative. And the first signaling pathway that was looked at is the PD-1 pathway. And PD-1 is a immunoglobulin-like molecule that has a CD3 domain. These immunoglobulin-like molecules usually bind to their ligands near the CD3 domain. And it was shown that PD-1 was actually upregulated on exhausted T cells in the LCMV model and subsequently in the HIV and SIV model. If you block PD-1 signaling by blocking its interaction with its ligand, which is ligand 1, then you can enhance the function of HIV-specific CD4 and CD8 cells, and it allows them to proliferate. The signaling pathway of PD-1 has actually been really well worked out. So when a T cell receptor is expressing PD-1 and it signals through the antigen-presenting cell, you get phosphorylation of the T cell receptor. Signaling through PD-1 actually recruits a phosphatase called through its SHP2 domain, and this is because the cytoplasmic domain of PD-1 has an ITIM and ITSM inhibitory motif which binds to these phosphatase enzymes. The phosphatase then is in vicinity of the T cell receptor and will block signal 2 phosphorylation signaling after T cell receptor engagement. There have been a couple other mechanisms that have been shown how PD-1 works. The other mechanism that PD-1 has been shown is that when you signal through PD-1, you increase this transcription factor called BATF. And BATF somehow decreases IL-2 production in the CD8 T cell. No one knows how PD-1 actually increases BATF transcription. And there was another recent paper published showed that PD-1 positive cells are sort of paralyzed. They're unable to move throughout the body, so they seem to have an abnormal chemotactic activity. So when you look in HIV infected individuals, we find that the PD-1 expressing cells have a decreased ability to proliferate, and they have an enhanced apoptosis. However, if you stimulate them with peptide, they still produce cytokines. So we feel that PD-1 is probably a marker of early exhausted cells, because the cells still have some preserved function in that they're able to produce cytokines. And Rafi Ahmed had shown in the LCMV model that there's at least two different populations of exhausted cells. There's one population of exhausted T cells that can be reversed by blocking PD-1, but there's another population of exhausted cells that seems to be even more exhausted that you can't reverse with PD-1 blockade. So we started looking at another immunoglobulin family molecule called TIM3. It has a similar structure to PD-1. TIM3 is, also has a mucin domain. That's why it's called T-cell immunoglobulin and mucin-containing domain. We don't know what the function of this mucin domain is. The cytoplasmic region has some tyrosine phosphorylation site, meaning that it can be phosphorylated somehow. But there are no ITIM or ITSM or ITAM motifs on this cytoplasmic domain. The ligand for TIM3 is this protein called galectin-9, and it has two regions on this protein that have a linker region, and these two regions are carbohydrate binding domains. So like most lectins, galectin-9 binds to any molecule that has glycosylated sugars on that is glycosylated or has sugars on it. So galectin-9 probably binds through these CRD domains onto these glycosylation sites on TIM3. TIM3 was first described by Vijay Kucherov because he was trying to find a marker for Th1 cells, and he found this antibody that bound to Th1 cells and described TIM3. So TIM3 is expressed on Th1 cells. The ligand for TIM3 is galectin-9 and also phosphatidylserine. When you add galectin-9 to a TIM3 expressing cell, it will actually die, and through apoptosis. Interferon alpha and interferon gamma can both induce galectin-9. So when you look at CD8 cells from HIV-infected individuals and CD4 cells, you find that they are actually have upregulation of TIM3 expression compared to uninfected individuals. This is using a polyclonal antibody that's very sensitive in detecting TIM3, and we see much more expression in HIV-infected individuals. If you take blood from one infected individual and you look at TIM3 expression on different virus-specific cells, you see varying levels of TIM3 expression. So we have one patient that has a T-cell response to CMV, EBV, and HIV. And if you gate on the tetramer-positive cells, you see more TIM3 expression on the HIV-specific cells compared to CMV and EBV. This is sort of summary data looking at a number of patients comparing TIM3 expression on different T cells against different viruses. And we find, for example, with influenza-specific viruses, we don't see much TIM3 expression on the T cells, whereas with HIV, we see higher levels of TIM3 expression. Surprisingly, we see in some patients low levels of TIM3 expression, and this probably relates to their functional capacities. Now, TIM3 expressing cells, so if you take these CD8 cells taken out of the body and then stimulate them with HIV gag antigen or a mitogen, and then look at cytokine expression or TNF-alpha or interferon gamma, you will find that when you gate on the TIM3 positive cells, there is very little cytokine expression. If you sort TIM3 positive cells into TIM3 positive component and negative component and then stimulate them with a mitogen and look for proliferation, these cells don't proliferate either in vitro. And of course, if you block the TIM3 pathway with an antibody to TIM3 called 2E2, you can enhance the proliferative capacity of HIV-specific T cells in vitro. We then wanted to look at see whether TIM3 positive cells are defective in their ability to kill HIV-infected cells, since that's the main function of CD8 T cells. So the first thing we wanted to look at was, well, do TIM3 positive cells, do they actually express perforin? And to our surprise, TIM3 expressing CD8 cells express more perforin than TIM3 negative cells. And this is sort of the summary data looking at either MFI or a number of perforin expressing cells. So TIM3 positive cells have more perforin than TIM3 negative cells. If you actually look at HIV-specific cells using tetramer staining, you see the same pattern where the HIV-specific cells that are TIM3 positive express more perforin than the tetramerase positive cells that don't express TIM3. This was a bit of a surprise to us. If we look at chronic progressors who have high levels of TIM3 expression, what we did is we incubated them with GAG peptide, and then in the presence of an antibody to TIM3 or an isotype control. And we found that if you add the antibody to TIM3, the CD8 cells will release more perforin into the supernatant compared to the isotype control. We didn't see this in long-term non-progressors because their CD8s don't express TIM3. Then we wanted to see whether these, what would happen if we block TIM3 and then see whether these CD8s can actually clear HIV-infected CD4 cells in culture. So these are cultures where CD8s are co-cultured with infected CD4 T cells. And then after a number of days, we look to see how many surviving CD4 T cells are present. So just looking at up here, with the isotype control, we see a large percentage of CD4 cells still expressing HIV P24 antigen. But when you add TIM3 antibody, the CD8 T cells are able to kill the CD4 cells and reduce the amount of infected CD4 cells in culture. We mainly saw this effect in chronic progressors. We saw very minimal effect to no effect with elite controllers because they don't express TIM3. Another way of determining cytotoxicity is looking at granzyme function. And there's an assay that you can use where you add a substrate and the granzyme, when granzyme is released and it affects the substrate, it fluoresces. And in this situation, in this one patient, you can see that when you add TIM3 antibody and co-culture CD8 T cells with targets, CD4 targets that have this granzyme substrate as well as expressing, that are transfected with the HIV GAG, you can see that blocking TIM3 enhances the ability to produce, to cleave the granzyme substrate. In addition, TIM3 CD8 positive cells also seem to have a defective ability to degranulate as measured by CD107A expression. So the TIM3 positive cells don't express as much CD107A after stimulation compared to the TIM3 negative cells. And if you block TIM3 with a TIM3 blocking antibody, you can rescue this defect in degranulation and increase CD107A expression, as shown here. So we think that the TIM3 positive cells have these following features. They don't produce cytokine. They don't proliferate well. There's an enhanced tendency to become apoptotic. They have a decreased ability to kill due to an inability to degranulate. These cells express high amounts of granzyme and perforin, but they just can't release them. So they're sort of constipated. Interestingly, TIM3 is not directly induced by HIV, neither does LPS enhance TIM3 expression. But if you take CD8 T cells and put them in culture and stimulate them with CD3 and add gamma C chain cytokines like IL-2 and IL-15, you will see TIM3 being upregulated in culture in about 2 to 4 days. So we were still puzzled on how does TIM3 work? How does it cause this abnormal, sort of, quotes, dysfunctional phenotype? And since there are so many abnormalities associated with T cell function, we thought that maybe TIM3 must work very early in the signaling pathway. So this is a diagram of how CD8 or just T cell signaling works in general, where after engagement of the T cell receptor and the CD4 molecule or the CD8 molecule, you get this phosphorylation of the zeta chain of CD3, and then you get this branching signaling pathway going through multiple branches. The first signaling event is probably LCK phosphorylation. And this LCK phosphorylation probably starts the whole process of signaling. So early on, you see LCK activation. Later on, then you start seeing ERK phosphorylation, MAP kinase phosphorylation, and FKB production. The other sort of general rule about T cell signaling is that phosphorylation enhances the signal to go downstream, and dephosphorylation with phosphatases probably quench or stops the signal. That's sort of a general rule. And you can look at T cell receptor signaling by looking actually at the interface between the T cell and the antigen presenting cell by looking at the immune synapse. So one way of looking at this is taking a T cell and a B cell, pulse the B cell with antigen, and then look at the immune synapse. And this is sort of a typical diagram of what the immune synapse looks like when you stain for CD3. You get this very tightly focused staining for CD3. So we did this with TIM3 expressing cells that were induced in culture. So the TIM3 cells were co-incubated with SCB pulsed B cells. And this is an immunofluorescent stain showing the nucleus is blue, green is TIM3 staining, and yellow is phosphotyrosine. So any signaling molecule. And what you can see is that of these three cells, two cells have lots of TIM3 in them. This cell doesn't have any TIM3, but this cell has lots of phosphotyrosine. So it seems like the TIM3 expressing cells, you don't see any phosphotyrosine in them at all, whereas the ones that have lots of phosphotyrosine, you don't see much TIM3 expression on them. We wanted to know, is TIM3 actually being recruited to the immune synapse? So one way of doing this is lysing TIM3 positive cells, and then you can actually do a sucrose gradient on that lysate, and then the very top layer will have your immune synapse, because the immune synapse is enriched in things called lipids, cholesterol lipids or lipid rafts. And the lipid raft component is always in the first eluate off of the sucrose gradient. And here's an example of the fractionation where number one is lipid raft, and you can prove that it's lipid raft because they usually contain GM1 ganglioside. And if you do a Western blot on these fractions, you find that TIM3 is found in fraction 1, which is the fraction that contains the lipid raft. And if you do immunofluorescent staining, you can see that TIM3 is green and CD3 is yellow. You can see that CD3 co-localizes with TIM3. So you see TIM3 actually at the immune synapse. Now we wanted to see, generally speaking, in T cell signaling, a good immune synapse is one that looks very clear and distinct and is usually associated with signaling. A bad immune synapse is one that looks kind of fuzzy. And what we found is that when TIM3 was present at the immune synapse, the CD3 staining was very fuzzy. When TIM3 wasn't present, then the immune synapse was very well seen. Here's an example of immunofluorescence where we see a good immune synapse here, where CD3 is present. And TIM3, actually, you probably can't see it, but TIM3 is actually in this cell, but it's been excluded from the immune synapse. And this cell is actually quite functional, because this red stain is for phospho-ERK. If we do this sort of experiment and add TIM3 antibodies into the system, we find that when you add the TIM3 antibody, 2E2, we see more well-formed immune synapses in immunofluorescence. This experiment is trying to see if TIM3 is at the immune synapse, is it doing something to LCK? And what we found with this Western blot is that TIM3 actually gets phosphorylated when you add galectin-9 to the system. So galectin-9 seems to phosphorylate TIM3. Also, TIM3 seems to, if you do a co-immunoprecipitation first with TIM3 and then stain for LCK, TIM3 seems to immunoprecipitate LCK. Now, there are two phosphorylated forms of LCK. There's an active form of LCK, which is important for T cell signaling, and that's called the tyrosine at 394. And if LCK is phosphorylated at tyrosine 505, it actually becomes inactive. And we find that TIM3 seems to be co-immunoprecipitating the inactive form of LCK at the synapse. So this is a diagram of the LCK protein. And in essence, it has two phosphorylation sites. There's an internal phosphorylation site, and that's the 394, and then this is the 505 C-terminal phosphorylation site. If this site is phosphorylated, it allows folding of this LCK molecule and prevents phosphorylation of the active site. There are certain kinases that are present that actually innately phosphorylate this site and make it inactive. CSK is one of them, which is actually found throughout the cytoplasm of the cell. So in this diagram, what we're showing you here is you were using an antibody to the active form of LCK. Yellow is CD3 to look at the synapse. Red is the active form of LCK, and TIM3 is green. And what we find is that when TIM3 is actually present at the immune synapse, the active form of LCK is not let into the synapse. So we're wondering, why is the inactive form of LCK not let into the synapse? So we're wondering, because we know that TIM3 does not have an ITAM motif, so is it recruiting any other molecules? So what we did is we did another TIM3 immunoprecipitation in the presence of galectin-9, and then we wanted to see, is there something that galectin-9 is binding to on these CD8 T cells when you also have TIM3 present? And we found two very common phosphatases, CD45 and CD148. They actually co-immunoprecipitated with galectin-9 and TIM3. And if you do immunofluorescence staining to TIM3 and CD45 and look at the immune synapse, we find that when TIM3 is present at the immune synapse, so is CD45 and CD148, two phosphatases that are that may dephosphorylate. So again, just showing you the two forms of LCK. And we find that if you add galectin-9 and do Western blot, and then immunoprecipitate TIM3, we find that galectin-9 enhances the ability of TIM3 to bind to LCK and to CD45 compared to when galectin-9 is not there. So this is what our this is what we think is how TIM3 is signaling. So here you've got the APC. Here's your T cell receptor complex. Here's CD45, which actually has a phosphatase, and here's the CD8 molecule that has LCK. So when you get T cell receptor engagement, then CD8 gets recruited as well, and then you have LCK there. LCK gets phosphorylated, and then LCK phosphorylates the rest of the active form of LCK will phosphorylate the rest of the T cell receptor complex, and then you'll get downstream signaling. Now, when you have TIM3 and galectin-9 in the system, so here's galectin-9. Remember, galectin-9 has two CRD domains, so it can bind to one glycosylated protein and bind to another one, bringing them close together. So what we think happens in this situation is that if TIM3 is brought into the immune synapse, galectin-9 will also recruit CD45, which is a phosphatase. And that phosphatase then will dephosphorylate LCK, the active form, and then prevent downstream, will dephosphorylate the T cell receptor and prevent downstream signaling. That's sort of our working model. So the working model is that galectin-9 binds to TIM3, it gets phosphorylated. CSK will phosphorylate the inactive LCK form. CD45 then will also be recruited and then will be involved with dephosphorylating the rest of the T cell receptor. Now there are other inhibitory molecules that are also been described, and this is a whole list of them, and they've been described in HIV and HCV infection. It seems that if you co-express these molecules, you have more immune dysfunction. And one interesting thing is that if you actually block a number of these receptors, that as Raffi did, if you block both PD-1 and TIM3, you can enhance the function of these T cells synergistically in the LCMV model. So our model of T cell exhaustion is essentially early exhaustion. You see high levels of PD-1, where I think TIM3 is probably marking later exhausted cells. And you also, when you have late exhausted cells, you also have these other markers also co-staining them. Now, why is TIM3 and PD-1 upregulated in these chronic infections? It's very possible that the reason why they're upregulated is because you already have immune escape, and maybe these T cells just can't eradicate the virus anyway. And as a result, the exhaustion markers are upregulated to prevent immunopathology. That's one hypothesis. Another thing that that has been found is that all of these exhaustion markers can also be upregulated during an acute virus or an acute infection. So PD-1 and TIM3 are also upregulated during acute phases of the infection. PD-1, because it always has a phosphatase, is probably down-regulating excessive immune activation during the acute phase of the infection. But I think TIM3, when it's upregulated during acute infection, might actually be a co-stimulator and may actually be enhancing the immune response. And there was a recent paper that was published in JI showing that in acute listeria infection, TIM3 actually may be a co-stimulator. And again, I think TIM3's function is probably dependent on the other molecules that are present on the surface near the T cell receptor complex, as well as the phenotype of the T cell. So with regards to clinical trials, there are a lot of clinical trials now that have been published on PD-1 blockade, particularly in the cancer literature. Here's an example of metastatic melanoma that improves after PD-1 blockade, and lung cancer that's improved with PD-1 blockade. Only about 20 to 30% of metastatic cancer patients appear to respond to PD-1 blockade, and it correlates with the presence of PD-ligand-1 expression in the tumor. So if you have PD-ligand-1 in the tumor, you may be a candidate for this type of therapy. The one caveat of blocking these molecules, however, is that it puts you at risk of further autoimmune pathology. And there was a study looking at PD-1 ligand knockout mice. When they get LCMV, when you infect them with LCMV, they clear the virus, but they don't survive. So they're able to get rid of the virus, but they get severe CD8 immunopathology, which actually attacks vascular endothelial cells, and they actually go into shock. So the operation was a success, but the patient died. So this is one thing that has to be taken into concern when you're thinking about these blocking these co-stimulatory molecules in chronic virus infections. So a lot of this work was done by Brad Jones and Kira Clayton, and a lot of students coming through the lab, as well as work with Doug Nixon and Lish Nolovu.